The COVID-19 pandemic is gripping the world's attention. As of April 18th, there are more than 2.2 million confirmed cases worldwide, and in the United States alone, over 33,000 people have lost their lives to the coronavirus disease. Currently, there is no vaccine, effective antiviral therapy, or treatment for COVID-19. COVID-19 is the respiratory disease caused by the virus SARS-CoV-2. The virus binds to a receptor on the body's lung cells and triggers a dangerous overreaction of the immune system called a cytokine storm. Cytokines are hormone-like messengers that activate the immune system. Researchers believe that the cytokine interleukin-6 or IL-6 triggers the hyperimmune response, causing the death of patients suffering from COVID-19. This video will explore how the drug tocilizumab may be used to target IL-6 receptors and potentially tame the cytokine storm caused by COVID-19. When the COVID-19 pandemic began, physicians in Wuhan, China noticed that critically ill patients were spewing dangerous levels of IL-6. So, they conducted a retrospective clinical study of 150 patients with severe COVID-19. They found that patients who died had higher levels of IL-6 in serum than those who recovered and were discharged. This suggested the cytokine storm was led by IL-6. Here I am illustrating the structure of the cytokine IL-6 in Pymol. Notice there are 4 alpha helices that make up this protein. IL-6 binds to and activates immune cells like neutrophils, macrophages, cytotoxic T cells, and B cells. In the classic pathway, IL-6 binds to IL-6R. Notice R stands for receptor. The complex is bound to GP130 on the cell membrane of the immune cell, triggering downstream signal transduction and gene expression. This will eventually amplify the immune response to the virus. In the trans-signal pathway, IL-6 binds to a soluble receptor called SIL6R. This complex will also bind to GP130 and initiate the same immune response. In the trans-presentation pathway, IL-6 will similarly bind to SIL6R. However, SGP130 will bind to the complex and prevent it from binding to GP130. This inhibits the immune response. Tocilizumab binds to both IL-6R and SIL6R, inhibiting both the classical and trans pathways. Recently, a clinical trial for the treatment of COVID-19 with tocilizumab was conducted in China. All 21 patients in the study met the criteria for severe or critical COVID-19, and they all exhibited elevated levels of IL-6 before treatment. After treatment with the drug, 75% reduced their oxygen intake, and 1 no longer needed oxygen. The proportion of peripheral blood lymphocytes returned to normal in the majority of the patients after treatment. There were, however, several limitations to the study. For starters, the study did not report the level of IL-6 after treatment with tocilizumab. Therefore, it is unclear if the drug effectively decreased IL-6 levels in serum. The small sample size decreases its statistical power as a study. Lastly, some patients who exhibited only shortness of breath were considered severe, while those who were admitted to ICU with organ failure were considered critical. The study did not specify how many of the 21 patients were suffering from severe versus critical COVID-19. Moving forward, the next steps should entail expanding the sample size population and ensuring the randomization of subjects to prevent selection bias. Future clinical trials should produce experimental and placebo groups with similar demographic characteristics, such as age, gender, and pre-existing health conditions. This would minimize confounding variables and produce more reliable data. Following the release of this study on 21 patients in China, Italy launched TOCIVID-19, a clinical trial with more than 300 participants in March, to evaluate the effectiveness of tocilizumab in the treatment of severe COVID-19. On April 5th, Denmark followed suit, beginning their own clinical trial on tocilizumab with 200 participants. And this past March, the FDA in the United States approved a clinical trial called COVACTA to evaluate the efficacy of tocilizumab in hospitalized patients with severe COVID-19. The manufacturer of tocilizumab declared they will provide the United States with 10,000 vials of the drug. Intensive care units around the world are overwhelmed by patients suffering from COVID-19. Since the 1918 flu pandemic and 2003 SARS pandemic were linked to cytokine storms as well, it is critical to understand why coronavirus patients are betrayed by their own immune systems. Since there is no available vaccine or effective antiviral therapy, developing an effective anti-cytokine therapy like anti-IL-6 is important for taming the cytokine storm caused by COVID-19.